Assalamu alaikum, good afternoon and good morning or good evening, depending on the world or where in the world you are right now. Welcome to the 10th webinar of the Future Belongs to the Tropics webinar series. This webinar series celebrates the tropics and highlights the importance of science and technology in the region. It is also a lead up to the International Conference on Tropical Sciences, TropSc, that will be jointly organized by the Mahathir Science Award Foundation and the Academy of Sciences of Malaysia in October 2021. The Mahathir Science Award Foundation is a non-profit seeking to encourage excellence in tropical scientific research that will bring direct benefits to countries in the tropics and the global community at large. Annually, we give out the Mahathir Science Award in recognition of the best scientific contributions and innovations to solving problems of the tropics in 4 different categories, namely tropical medicine, tropical natural resources, tropical agriculture, and tropical engineering and architecture. The Academy of Sciences of Malaysia is an independent organization representing the scientific minds of Malaysia. It carries out strategic studies that are independent, evidence-based, reliable, and timely for the benefit of all. Today's webinar is a joint effort between the Mahathir Science Award Foundation, the Academy of Sciences of Malaysia, and the International Science Council Regional Office for Asia and the Pacific. Today, we are celebrating the International Day of Biodiversity, and we will focus on the future of Malaysia's biodiversity data. We have an excellent lineup of speakers today, and we hope that all of you will come out of the webinar having learned a few new things. Now, without further ado, I'd like to invite the chair of our upcoming TropSc conference, Professor Emerita Maslan Othman, to give her welcome remarks. Please.
Thank you, Alia. And good day, everyone. Welcome to our webinar series entitled The Future Belongs to the Tropics. This webinar series is a lead up, as you could see, to the International Conference on Tropical Sciences that will be held on the 24th to 27th of October. And it's this year is co-organized by the Mahathir Science Award Foundation and Academy Science Malaysia. If you have been following us in the past, you will have noticed that through this webinar series, we have explored the subjects of mangroves, youth, health, mountains, cities, forests, and even coffee. Today, we will discuss a topic almost synonymous with the tropics, biodiversity, because we are celebrating the International Day of Biodiversity. I know you're all converted here, but just to remind ourselves, the tropics host approximately 80% of the world's biodiversity. Tropical forests host over 2/3 of the terrestrial biodiversity, despite covering only 7% of the global land surface. Similarly, tropical coral reefs, despite covering only 0.1% of the ocean floor, support 1/3 of the marine species. So yes, we all know why we are here today. Now, at the heart of the tropical Asia is one of the world's mega diverse countries, Malaysia. Malaysia is home to an extraordinary number of species, many yet to be discovered and identified. Now, the wealth of species naturally translates to a wealth of data. And currently, the data resides in many collection centers all over the country. Now, how can Malaysia move forward in consolidating this data so that it serves the national interest? So at this point, I should say that even though today we're talking about the Malaysian national interest, I am sure, I'm very confident that many of these issues are common throughout the nations of the tropics. So hence today, we will follow on from the discussions initiated actually 4 years ago at a workshop called Naming Nature. I know many of you, in fact, maybe the majority of you, are from the first Naming Nature event. This workshop gathered some of Malaysia's most experienced taxonomists, whom I hope are again here today, with a group of young, enthusiastic researchers. Now at that meeting, they clamored for a system to better handle identification, digitization, and communication within the community. So today's webinar, which we've decided to name Naming Nature 2, will address these same issues that have remained unresolved for 4 years. So we have with us an impressive lineup of speakers representing different stakeholders in Malaysian biodiversity, and I look forward to the discussions that will ensue. And just to give you a heads up, halfway through the discussions, the question and answer within the panel of the panelists, we will be throwing out a poll for you to answer. So please answer that poll. So now I leave you in the hands of our esteemed moderator for the day, who, together with our distinguished keynote speaker and panelists, will collectively help to craft a future path for our Malaysian biodiversity data. So, Prof. Nohaiti Ahmad, she is a herpetologist at University Kebangsaan Malaysia. I've always known her as the snake person. I'm always scared of snakes, so she's the one, who shows me how silly I can get and how brave she is. So over to you, Nohaiti. Thank you.
Thank you very much, Datuk Chairman, for the introduction, and hello everyone. I'm so honored to be here with you. As you know, the United Nations has proclaimed 22nd May as the World International Day for Biological Diversity to increase understanding and awareness of biodiversity issues, and that's why we are here under Naming Nature 2. Specifically to discuss on the way forward towards a consolidated biodiversity database for Malaysia. This is also in line with the overarching theme of the Biodiversity Day 2021. We are part of the solution. Nature-based solutions can offer ways to promote human well-being, tackle climate change, and protect our living planet. As Datuk Maslan has already mentioned, we have gathered strong panelists that will cover topics on policy, digitization of specimens, collections, taxonomy, and database. And as part of the global megadiversity country, biodiversity and taxonomy work is vital, and collection is a must for every scientist. And deposition of specimens, I would say, is a challenge. For biodiversity, taxonomy is often dubbed the old-fashioned discipline or stamp collecting, and but in actual fact, it's fundamental to modern biology, requiring intensive fieldwork, information technology, and sophisticated labs, and should not be downplayed. It's more critical today because most species of biodiversity still await discovery. And in Malaysia, we have at least 22 depository centers, and among others are the Center for Insect Systematics, or CIS, in University Kebangsaan Malaysia, which is one of the largest collection of insects in Southeast Asia, with over 100,000 specimens, in which more than 120 are type specimens. And then we have the UKM Herbarium also has more than 120,000 specimens, of which 50,000 seed plants specimen. We also have Forest Research Institute of Malaysia, or FRIM, which is another major national herbarium and arboretum in the country. And on Malaysian biodiversity database, we will have the perspectives from the government agency, from the private sector, and the academia. So let's hear more from each panelist on how they establish their respective databases and how they work together to achieve a consolidated mechanism for sharing the database to the public. So this webinar is timely to update us all on the status, issues, and the way forward. First off, let me introduce you to the first speaker, who is going to present the plenary, Dr. Khairul Naim Adham, who is the Undersecretary of the Biodiversity Management Division, Ministry of Energy and Natural Resources in Malaysia. Dr. Naim, you have 50 minutes, 1 minute left after you hear the first ring, and your time will be up after 2 rings. So without further ado, I would like to present Dr. Naim. You have the floor.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and a very good afternoon. Happy International Day of Biodiversity, and I think not too late also to wish all of you Selamat Hari Raya. Thank you, Prof. Nur Hayati, for your kind introduction. Yang berbahagia Professor Emeritus Datuk Seri Dr. Maslan, distinguished speakers, participants, ladies and gentlemen. First and foremost, I would like to thank the organizers for inviting me to this very important session. And it is my pleasure to be entrusted to deliver a keynote speech. I have a number of slides to be shared with participants. Can you see my slide?
Yes.
Okay, thank you, Professor Moderator. In line with the topic of our webinar, my presentation will be focusing on a national biodiversity agenda from the perspective of biodiversity database in Malaysia. On introduction, it has been also highlighted by Prof. Nur Hayati. I will share again about the richness of our biodiversity in Malaysia, followed by the key issues and challenges in managing and conserving our precious biodiversity. We will be looking also at the policy directions at global and also national level. A few national initiatives supporting the consolidated biodiversity database, and finally the way forward. Ladies and gentlemen, Malaysia is one of the world's mega diverse countries. It means Malaysia holds at least 70% of global biodiversity. Prof. Dr. Maslan mentioned about the richness of tropics. I will be more focusing on the richness of our country, Malaysia. Based on National Biodiversity Index, Malaysia is ranked 12th in the world. If you can see on the slide, Malaysia's flora and fauna is rich and diverse, where not many countries in the world has this kind of biodiversity richness. Malaysia has over 15,000 species of flowering plants, hosts more than 300 species of mammals, over 500 species of reptiles, and almost 800 species of birds. Our marine and coastal biodiversity is equally amazing, with more than 1,600 marine fishes. Being a mega diverse country is great, but managing our precious biodiversity is indeed challenging. It means we must work very hard to catalog and then conserve our biodiversity. What are the key issues and challenges? Among other key challenges is several inherent issues, such as segmented jurisdiction between states and federal governments, as well as between implementing ministries and agencies. Under the federal constitution, as you can see on the slide, under Article 74 to 79, natural resources, including biodiversity, appear on the state list, whereas Sabah and Sarawak and Borneo are granted greater autonomy on managing their natural resources. In addition, the rapid socio-economic development in Malaysia has exerted pressures to our biodiversity. There is also a limited capacity and capability for managing effectively our biodiversity conservation, including human resources as well as financial resources. Last but not least, on the right-hand side, as you can see, it is about a biodiversity database. For a mega-diverse country, comprehensive biodiversity database is crucial for effective monitoring, evaluation, and reporting. Inaccessible and fragmented data has always been an issue in managing our biodiversity due to different authorities and jurisdiction collecting and managing the data. Moderator has highlighted that UKM has huge number of specimens, insects, and also herbarium. We have also many other research institution and universities that hold a large collection of records in the form of specimens, samples, germplasm, genetic information, etc. It was estimated that we have more than 3 million scientific collections from the 22 collection centers that have been mentioned by Prof. Moderator just now. This has complicated efforts toward producing a comprehensive biodiversity database, as we need a comprehensive database to enable us to determine priorities for interventions. Issues related to sharing of data can be classified into two. Yeah? The first is data is not available. Secondly, data is not accessible. CBD has highlighted the concern of on sharing data, and they have identified 4 key barriers for sharing data. The first one is psychological and behavioral barriers. Second is barriers relating to describing data, practical barriers, and inadequate strategies and resources. I will give a number of examples for each of the barriers. Psychological and behavioral barriers is referring to unwillingness to share data for a number of reasons, including concern that someone else will get the benefit, or they misuse the data and gain profit unfairly. Under this barrier also, it includes barrier related to laws and regulations protecting copyright. Now let's look at barriers relating to describing data. It includes the use of many different languages, taxonomies and classification system, metadata standards that make the cost of complying to certain requirement is too high. Practical barriers include data that is not available in digital forms or formats, problems of access to technology, limited information storage, and the inability of data integration. Finally, on inadequate strategies and resources, this include the lack of strategies and resources for addressing the demands of stakeholders, because database development often needs more funding, and it's often we develop a database without sufficient resources. Now let's look at the policy directions. At the global level, Aichi biodiversity target biodiversity under biodiversity strategic action plans for a period of 2010 to 2020 recognize CHM, clearinghouse mechanism, as one of the tools to widely share biodiversity information. For your information, CHM is an online information exchange platform of the CBD, Convention on Biological Diversity, that will encourage the sharing of information, and the information can be used for better planning, implementation, and reporting, both nationally and internationally. Establishing CHM is one of the requirements under the CBD, and Malaysia is a party to CBD since 1994. Article 18 of the CBD stated that the need to establish a clearinghouse mechanism to promote technical and scientific cooperation. In addition, CHM is also important as a tool for public education and awareness. It also helps in accessing the transfer of technology, exchange of information, and also for reporting purposes. Aichi biodiversity target will be replacing by post-2020 global biodiversity framework. We are in the midst of negotiating the document. Under this zero draft, target 19 specifically proposed of having a database with a quality information that will assist in effective management of biodiversity. Now let's look at the policy direction at the national level. A 12th Malaysia Plan, a national development plan, 5-year national development plan has outlined the importance of having national platform for the collection and sharing of comprehensive biodiversity information. This is a clear indication of the importance of having comprehensive biodiversity in order to achieve sustainable development. Mampu has implemented the Open Data Ocean Empowerment Program in 2014 to create and promote an open data ecosystem. And as in the final box, NPBD is referring to National Policy on Biological Diversity. That will be further emphasized in the next slide, provides a clear direction for having a comprehensive biodiversity database for effective management of biodiversity. In a GIS, National Policy on Biological Diversity has 5 goals, 17 targets, and 57 actions. And the ultimate goal is to achieve the 3 key objectives of the CBD, which is conserving biodiversity, promote its sustainable use, and ensure fair and equitable sharing of the benefits. As highlighted in the red box, there are at least 7 targets that support the establishment of comprehensive biodiversity database, namely target 3, 6, 9, 10, etc. Target 16 specifically highlighted the importance of establishing comprehensive databases and monitoring programs. Under this, there are at least 6 key actions, among others, continue our efforts on documenting a nation's flora and fauna through the enhancement of taxonomic work. Probably the next speaker will be explain further on this action. Strengthen the national clearinghouse mechanism while ensuring that information can be accessed and shared by scientists, resource managers, and other stakeholders. Target 16 also highlighted the importance of improving knowledge and link between climate change and biodiversity, and improving the interface and communication between science and policy. I will share this slide to participants so that you can get the detail of the actions under NPBD. And many other actions under various targets that I have mentioned earlier. Probably we can discuss this afterwards since the time allocation is very limited. Now we look at our national initiative. I would highlight 3 key initiatives, namely clearinghouse mechanism. Malaysia has Malaysia Biodiversity Information System, or in short, MyBIS, a one-stop repository for biodiversity information in Malaysia. MyABS, Malaysia Access to Biological Resources and Benefit Sharing as a platform to implement Access for Benefit Sharing Act, and My Biosafety to promote high standards of biodiversity in the research and biomedical community. On the second bullet point, Malaysian Open Science Platform was launched on this November 2019 to develop a trusted platform that makes raw research data findable, accessible, interoperable, and reusable. This probably will be also explained further by the next speaker. And thirdly, Malaysian Oil Palm Genome Program to facilitate the development of important genomic platforms to provide a deeper understanding of genome architecture and resource for biomaker discovery. Okay, and my last slide is on the way forward. The theory of change is that we need a national level policy to build consistency and coherence. Without national level enabling frameworks, the potential to increase synergy among various stakeholders may be missed. We need to avoid silos and reinvented wheel. And the policy must be further supported by guidelines for sharing data. There is also a need to inventorize our existing biodiversity database to avoid duplication of effort in data collection and management. The third point is on the institutional framework. It is timely for Malaysia to have a center of excellence in biodiversity management. We are in the midst of establishing Malaysian Biodiversity Center, where one of its key role is on strengthening and enriching biodiversity database in Malaysia. Last but not least, on capacity building to address severe lack of skilled bioinformatician. So I hope that by doing all these action, it will enhance Malaysia's reputation as not only a biodiversity-rich country, but also biodiversity database-rich country for better management of our biodiversity. With that, Prof, I conclude my presentation. Thank you.
Thank you very much, Dr. Naim, especially for keeping within your time limit. We will hold a question for you. There are many burning questions for you towards the end during the panel discussion. So we move on to our second panelist, who is going to present on the overcoming challenges in taxonomy research. Dr. Liu Thor Seng is a researcher at the Institute for Tropical Biology and Conservation in University of Malaysia, Sabah. Dr. Liu, you have the floor now. Please share your slide. Thank you.
Thank you, Prof. Nohayati. Thank you, Prof. Maslan, for the invitation to this amazing seminar. So today I'm going to talk about the challenges in taxonomy research, particularly focus on the knowledge and data management. So first of all, as I noticed that our audience, our participant is from the diverse background. So I think it's good to describe a bit what is a taxonomy research. And then I'm going to go to this talk about the taxonomy research, the challenges in terms of the knowledge and data management. And then last, to show some recent development which can help us to overcome some of the challenges. So first, taxonomy research. So basically, all of us are taxonomists to a certain extent, without you realizing it. So I give you an example. So I just described about the taxonomy workflow. So I think this is happen in the everyday life. We observe animal, plant. The next thing that we do, we try to learn more after we observe and try to know what is the species. This is the human nature. We want to know. And if we are lucky, the organism that we are interested, the knowledge, the taxonomy is well established. We can easily identify them just by referring to some secondary taxonomy reference or some popular field guide. So basically, this is also the starting of the workflow as a taxonomist. It's no difference. So basically, we start with observation and try to learn. So what made the difference is for taxonomists as a profession, after we recognize the species, we will go a bit one step further, which is to documenting the species record. So that means that we keep the specimen when necessary and also publish the record. It's very important to publish a record. So these are the example. And in many occasions, for some species that is less known, we will have some problem to identify the species. When we cannot identify the species, this is where the taxonomists need to step in and then do some serious work. So the first thing is to find, locate, get all the taxonomy resources. In this case, it's a taxonomy references and also the specimen. After getting all this resource, the next is to integrating all the information. Try to put all the puzzle together. Then the next step is to describe the species and revising the species group. So this is very technical, which I'm not going to talk about it. And then last, we're going to publish it. So this is how we improve the state of the knowledge of the biodiversity in around us. Okay, so the challenges, so what we did actually is still the same. We observe, we learn. So while we integrating the resource and revising the group, revising the species, we also learn at the same time. What is different is as a taxonomist, at the same time, we also contribute. We contribute to the existing knowledge, try to improve our understanding of the certain species group. At the same time, we also need to obligate to share our results, share our data. So what are the challenges? So actually the challenge is at this stage, okay? Which is related to today's topic, it's about the knowledge and data management. So the taxonomy is a data-rich science. So we have to deal with all the works that have been published, no matter how old they are. We have to locate all the specimens that have been studied before. So you can imagine over 100 years of research, all the specimens is scattered all around the world, and we need to spend a lot of time just to find them, not to mention to study them. This is the first step. At the same time, there's a mismatch between the taxonomy resources and also undescribed species. Most of the resources are in the centralized in the developed country, while most of the undescribed species is in the developing country, for example, in Malaysia. Last but not least, the information is being logged in the publication. Even though now we have the digital publication in form of PDF, but information is logged in that document. It's not easily accessible. It's very difficult to be integrated. So these are the main challenges which is related to today's topic about the data and knowledge management. So I give you an example. So this is all the species that can be found in Sabah. So we have 300 over species. And what you see here is the year of the description of the species. Just imagine that this cannot be done one person. It's done by all these giants. So actually we're standing on their shoulder. So during the learning process, when they integrate the specimen, they revise the group. Sometimes they are successful, okay? They left a legacy of the specimen that they collected, provided it's properly cataloged, we deposit in the museum, it's easy to find them. At the same time, for those taxonomy problems that they can solve, they will publish it, okay? So, but maybe not all the time the problem can be solved. So the workflow that I show you, sometimes it just stops halfway. That means that the knowledge that have been gathered and learned and integrated was not published. So this is an issue. So when you have the specimen and also the reference, so this is how it looked like. So this is in the old days, where this is an example of the species cards which developed by one of the taxonomists. Okay, it's used by one of the taxonomists. So what you have here, you have the species. And then after that, you just record all the reference that mention about this species. So you try to integrate the information for our own use. So you can imagine for every taxonomist, every researcher, they will keep some sort of the personal notes. However, this thing is not published because it's not conclusive, and it's most of the time it just left, it's just lost during the process. And even for the PDF, it's no difference. It's just compilation of the information, but all the information is still logged in the publication. That means that whoever who want to continue the study, they still need to go through the painstaking process to gather the reference, to allocate the specimen that deposit all around the world, and then integrate them again, then use it again. So this is what happened at least 10 years ago. At the starting of my career, fortunately or unfortunately, I have to experience this at the early part of my career. So how to overcome this? So basically, there's a very interesting development when I just start my career during my PhD. Okay, this is how it looked like in the past. So for the first 5 years, I worked in a very systematic and traditional manner. But now, from time to time, there are always a new tools and new facility. I've used the word facility that have been developed. Okay, for example, GBIF. So this is a database that facility that compile all the metadata and also species record for all the resources, including the museum collection. So at the very beginning, this database is not very useful because it's not comprehensive, it's not complete. So for example, until 2010, there are about less than 1 million museum record, okay, not including other records. But now there are more than 10 million records. So there are a lot of the exciting development in terms of the specimen. So specimen-wise now is very important, easy. You just key in the name, you can get the specimen. If you're lucky, you can get the image. And the next is about reference. So in 2007, or around that time, there's a biodiversity heritage library facility have been developed. At that time, when I start my work, there are only 300 titles available. But the platform is very useful. You just type in the name, they will search all the reference for you. And they even pinpoint to the particular page where you can just click and then read to the page. It's very useful. And now there are more than 160,000 titles available. So now everything is on your fingertips. Everything is available. If not 100%, maybe 80, 90%. It's good enough. It's very, very good. Then the next step is how to integrate the information. So it will be quite silly to use the traditional method, to use a species card or use the Excel to compile it ourselves. Actually, there are a few platforms that are available. So this is one of them, which I think is quite good. And also to help to link all these information together. So for example, rather have the species cards, you can key in all the information, and it's made our learning, even is without comprehensive conclusion, can be shared immediately. Okay, real time. We don't have to wait until the paper is published or wait until we have the conclusion. So for publications, there are new publication journal. For example, this is a very advanced. So when you publish, all the information will be sent to this facility. At the same time, they will also integrate the information for other facilities to help us to locate the information. So no longer logging into the publication. So this database has several characteristics. First, is accessible. There's no limit, no red line, nothing. It's very easy. Scalable. You can see the example. It's evolved so fast. Most importantly, is integrated. And since 2014, I think most of the database is integrated. So I started with this slide. So I have half a minute left. We start with the observation and learning. This is everyone do. I can imagine, starting from today, every person can become a taxonomist. So there's an app called iNaturalist. I think most of you are familiar. It's very useful. So for example, rather than, I think nowadays for future generation, the first thing that they do when they see the specimen, they will observe, and then they will take picture rather than look for the book. Okay, and this app, in this app, what the next thing that they, so in this app, the next thing that you can do actually is to ask the apps to help you to identify it easily. Okay, pay attention here. So after you take a picture, you can just click here. Then they will search, do some AI searching. They manage to find the closer one. If you are not confident, you still can compare with the record that deposited by the other user. So in this way, you can know what is the species. At the same time, once you have the name, for anyone, you don't need to be a taxonomist. You can just key in the name. You can get all the information that you need. Specimen, paper, knowledge of the taxonomist, if the taxonomist is making some database for themselves to share their learning. So basically in the future, everyone will be the taxonomist. They are not only observe, they also share, contribute when they upload all the information, and then learn by using all this existing information in the database. So that's all from me. So thank you.
Okay, thank you, Dr. Liu, for that very comprehensive presentation. And I'm sure we will have many questions for you later on. Next on the line is Mr. Lim Kui Fong, who is going to present digitizing Malaysian biological specimens. Mr. Lim is the founder and the managing director of BioVis Informatics Sendirian Berhad since 2009, and he's a bioinformatics specialist with a focus on taxonomic systems architecture. Mr. Lim, can you share, can someone please share Mr. Lim's slide? And then you can have the floor.
Okay, thank you, Dr. Nohayati, Prof. Good to see you again after a few years. Yeah. Thank you, Prof. Dr. Maslan and all, everyone for the invitation. So today, very briefly, I will quickly go through experience that I have with digitization of the Malaysian collection at the Natural History Museum for approximately 1 year, July 2017 to June 2018. So basically, I worked together with Mr. Lawrence Livermore, who is in part of the panel, to actually bring back our Malaysian collection in digital format. So the project was very successful. We managed to bring back close to, next slide please. Yeah. We bring back close to about 21,000 images, okay? So including more than about 5,000 Wallace related specimens, which are very valuable, I believe, because many of these include holotype, you know, and all the type specimen that is very useful for our local scientists to make research. This collection is now sitting in MyBis. So we have actually digitized them, brought it back, and then stored in our national biodiversity system, information system. Okay, so this is a quick breakdown of the collection that we have. Lawrence's team was very helpful. All right, without their cooperation, this was not possible. As Dr. Liu mentioned just now, okay, you know, not only just digitization, it's about, you know, locating the names and the families and then the nomenclature, right? When we bring back this digital collection, we're not only just bringing back the images, but also the related content, the metadata, for instance. And this is very valuable, especially when we want to do research. Okay, so next slide, please. Okay, so quickly, I just want to go through the adventure of how the NHM does the imaging. All right, I think most of you already, most of the universities already have this such setup. Okay, this is one of the two or three stations that the Lawrence team helped to set up for us to digitize the Malaysian collection over 1 year. Okay, we are talking about not only just one side of the specimen. We have to flip the other side, you know, especially for plants and insects. We have to flip the other side to get the both sides of the image, right? And sometimes you have to do the side imaging. So it's a lot of effort. So sometimes what call this? One specimen we are talking about 4 or 5 angles, all right? And we are talking about a very high resolution image. So the average image size that we have at the MyBis now is about 10 megabyte, 10 to 15 megabyte. And we can zoom in with detail. Okay, so we are talking about, if possible, to bring our imaging at this level. We should benchmark our imaging quality at this level, where we can zoom in to a very specific, very good macro levels, so that we can actually analyze, okay, and analyze each of the specimen more accurately. Next slide, please. So this is a setup. And once you set it up, and we just bring the specimen from the drawer, you see the lady there, okay, beside the white box, and you see a drawer there, right, in between the white box and the PC. This way, you pick the specimen from the box, and then you put it onto the light box, and then you adjust the camera on the mounting stand. Okay, next, please. And you'll notice that the specimen comes together with the tags, you know. Okay. Now, imaging specimen, in some cases where it is almost tens and hundreds of years old, require very, very careful handling. You see the tweezer up there? Okay. So it's not just plug a specimen, put it onto the foam, and then take picture. Okay. You have to unpin, all right, and then place the specimen on top of the foam, and then rearrange the metadata, which can come in a few pieces of paper. You can put it on the side or around it, and then you have put it up on a ruler, okay? And then only you take the image. So just to set up one specimen is not 1 minute job. It could take up to what, 10 minutes, depending how fragile the specimen is. Okay, so it is time consuming. It is not easy. So to do over 20,000 images with over a year, that was a feat, I would say. Okay, need full-time workers to actually do this, morning to night, every day. Okay, next please. This is how they take the specimen from the pin. Right, the pin is not removed from the specimen. Only the metadata tags are removed. And then the specimen is pinned onto the foam together with the ruler here, so that we can actually estimate the size. Right, next please. Do you see the metadata text on the right side? So basically, this is how it is done for each and every specimen that we took the image from at the NHM. Okay, next please. So this is how it looks like from top, okay, from the camera eye view. So when we did this exercise, I thought it was quite a simple, straightforward thing, but it's not, because just from here, you can actually obtain a lot of information from this specimen itself. The size, then the metadata tags that relates to the library of information found inside the content boxes inside the NHM. And you notice some specimens, they have a QR code already. Okay, so I doubt many of our Malaysian collection have QR codes or not. So this is another area that we should look into, because digital collection, right, with the QR code will be able to help the researchers fairly quickly locate where it is stored. Okay, so when we talk about digitization, we also have to look at digitization of the location of the storage of the specimen. If you want to really collaborate, between all the collection centers in Malaysia, I think this is one way we have to really, push forward. Okay, so in a nutshell, this is the experience that I went through at the NHM. Now, Dr. Maslan and her team are in the process of coming up with some guidelines, I would say. All right, talking about benchmarking and coming up with guidelines for researchers, not necessarily advanced researchers, maybe even school children, okay, or even newbies who just gone into the field of taxonomy. So there is a process involved with this, all right. The process is, as I took this from the Arkansas State University Herbarium, and I'm sure the NHM follows this process, okay. What we are saying is that I think in for Malaysia, it's like cowboy country lah. Everybody have their own rules and setup, okay. But to be able to kind of like set up a good benchmark, we need to identify a process. We may not necessarily follow the process of NHM or Arkansas State University, but I believe we should come up with a good guideline and a very robust process, which we can share for all the research centers and anyone who wants to do taxonomic research, right? So this is a process basically that outlines the process, the flow of digitizing an image. Okay, and this one here, I think Dr. Liu has already explained in detail. But if we can come up with a flow like this, I think it's very useful, not only for matured researcher, prominent researchers. We're talking about even newbies, students, okay, young researchers. If we can follow a guideline like this, then everybody is following a certain standard. Okay. Okay, next, please. In Arkansas State University, what they do is that they get a lot of students to help them do digitization. All right, they really make use of their students. Okay, so what they break down the work according that is run by all these researchers, and then they identify each student, how many work in pairs and how many work alone. Okay, and then imaging is one set of students, databasing another set of students, and you notice how many amount of recorded works is done by each of the student here. Okay, and then there's another process called uploading. And at the end of the whole process, one round, and you see how many specimen is done. Okay, so we have a certain process here that that will help in a way to document the flow and process. Next one. Okay, so once you have some sort of identification, how do you do the databasing, right? Many universities, I think they have their own system. Some uses Excel, some uses a system that they build by themselves. Sometimes, I even seen a process, a database built by FoxPro, which FoxPro don't even exist nowadays. Okay, so what happened is that many of these university systems, they are not online yet. They are offline. They're stored in their particular system. Okay, so once you store into here, right? Then later on, these are not confirmed. These are just observations. Then after more research, then after that you say, okay, let's put it up online, publish it on a reputable database, for instance, or even publish it in a journal. Okay, but this exercise actually helps the student to what call this get an idea of the flow. I think it's not so much about doing the work, it's about understanding, what is the benchmark required to do quality work. I saw a question in one of the chat. We're talking about, maybe it's the Dr. Nohayati asked this question, I don't know. We said, yes, we have nice images, but what about the quality of the content? Is it useful? Why is the usage of our database so low? Why? Okay, why we want to go to GBIF? Okay, so I don't have much time. Next one. Right, once you have all this data, then we can use it for distribution studies, which is very useful. Okay, next. And I think very important, acknowledgement. We don't do this all the time. Okay, we don't do this all the time. Even for students, we do not put their name for any contribution. We maybe put it up in once the journal is published. But during the digitization process, I believe and some sort of acknowledgement like this is very useful. Okay, so this is some ideas that I would like to the Academic Science of Malaysia and the University of Malaysia team to consider in terms of digitizing. So quickly, just to summarize, organize all specimen in cabinets using updated taxonomy nomenclature. Fine, right? We have to do this first. Without this, how can you collect the specimen and bring it out to scan, right? Then to digitize, you have to purchase the equipment. Very quickly, we need a camera, a light box, and very important, macro lenses. Macro lenses is so important. The body is just the body. You need good macro lenses, and it's not expensive to set it up. And a vertical stand to mount the camera. Okay, and image each specimen individually so that you have a good protocol. These are the setup when you do imaging. Those who are interested can download this slide. You can follow this setup, this settings up in your camera. Next one. All right, database. I showed you about the database from the Arkansas University. You can have your own database, but later on when you when you convert this data offline to a more bigger database like MyBeast, right? So you make sure that the views can talk to one another. That's very important. Okay, backing up data, very important. This is something I think not many universities pay a lot of attention to. Okay, backing up a few types. First is backing up physically, through the external hard disk, and also backing up online. Invest a bit more on cloud facilities. Not just talking about the Dropbox and all, but a good cloud facility. And finally, combine all to make it accessible to the scientific community. So of course, if it is good, if we can push up this content to GBIF, but this is an issue with our local researchers. So I think during this webinar, we can discuss further why we are not doing that. Okay, so that's my presentation. Thank you very much.
Okay, thank you, Mr. Lim. I hope you can stick around for the panel discussion later on.
Yes.
Next presenter, we are glad to have Mr. Lawrence Livermore from the Natural History Museum of London. You will be talking about overview of taxonomic backbones. Mr. Lawrence is a digital project manager with 5 years of experience at the Natural History Museum and specializes in digitisation, biodiversity, informatics, and data. Without further ado, Mr. Lawrence, please
Thank
start
you.
your presentation.
Can you hear and see me okay?
Okay, yes.
Yeah. Okay, well, thank you for inviting me to speak today. It's a pleasure to continue working with my Malaysian colleagues on your biodiversity data. So I wanted to start today by just talking about what a taxonomic backbone is. They're essentially a digital checklist, but with some additional important properties. One key difference between a checklist and the backbone is the interconnectedness with other systems. A national backbone would typically rely on taxonomic data from expert taxonomic community networks, such as some of the examples on the left. International communities can be used to fill in some of the gaps. Very few countries have specialists to cover absolutely everything, but it may also make sense for national experts to contribute their knowledge to international efforts. Many national backbones, for example, use data and contribute directly to the World Register of Marine Species, for example. And while you can start with nomenclatural data when you're kind of building your biodiversity systems nationally, this can of course be expanded in the future to include and link to literature, specimens, molecular data, and a range of other data linked to these names. And this interconnectedness makes it easier for other national stakeholders to make use of the data and allows for easier construction of future platform functionality. So on a fundamental level, taxonomic backbones provide you with a comprehensive, stable list of all species that occur in a country along with their status and synonyms. This alone is incredibly useful for scientific publications, other biodiversity databases, and for the organisation and labelling of biodiversity specimen collections, both living, so those in zoos, botanical gardens, or fungal collections, or those in preserved collections, herbaria, museums, biobanks. They allow you to estimate the diversity of particular taxonomic groups. That could be in the entire country or specific parts of the country, such as areas of conservation. And having a list allows you to identify knowledge gaps by looking at higher taxa for which species information is lacking, or where taxonomic or nomenclatural problems are highlighted in your national backbone. And of course, being digital and linked to other resources makes it easier to update and maintain the most up-to-date versions compared to published or static data sets of floristic or faunistic treatments. And finally, it allows legislators and regulators to use the correct name in documentation. For example, when dealing with alien, invasive, traded, cultivated, pest, harvested, or threatened species. So while perhaps obvious, it's important to recognize that understanding biodiversity requires us to look beyond national borders. A taxonomic backbone can form the starting point of a portal, but as I've said, this can expand to include eDNA, remote sensing data, literature, observations, digitized specimens from collections. But as we've heard from previous speakers, in order to answer some of those very big questions, when you're thinking about a taxonomic backbone, you need to link that backbone up to international data. And so to this end, publishing to and using data from the Global Biodiversity Information Facility should be a goal of any national taxonomic backbone. And building an internationally connected national taxonomic backbone will enable and support research on biodiversity and sustainable development in areas such as conservation, supporting and understanding the monitoring of protected areas, threatened species and invasives, In food security, by documenting crop-wild relatives, national genetic diversity, supporting the planning and impact assessment of fisheries, climate change, modelling different human impacts on species ranges, understanding their adaptability and what we may be able to mitigate, on issues such as human health, understanding the risk of disease based on the occurrence of vector species, their hosts and potential resources, medicinal plants and ethnobotanical knowledge, and hazards posed by biodiversity. You would need to know which snake has bitten you to get effective antivenom. All of these research areas are supported by the data aggregated through GBIF, who directly support biodiversity indicators under Aichi Target 19. This is crucial to reviewing international progress, the current implementation, and strengthening the future implementation and operation of the Convention on Biological Diversity. So, how do you build a taxonomic backbone? This presentation, this section could form its own presentation. So I've kept it fairly light, but there are three areas that I think you should consider when designing and building a national taxonomic backbone. And I would start with your stakeholder and user requirements. What do they need from the taxonomic backbone? How will you ensure it's useful to them? How will you build a community to fill taxonomic gaps in national knowledge? There are many technical requirements for a modern taxonomic backbone, with a lot of important functionality going on behind the scenes, essentially invisible to the users. It should be available in a machine and human readable open data repository with permissive licenses. It should use international standards and provide rich data. From a systems design view, should you use an existing platform? Do you want to self-host or externally host? And finally, you should consider the sustainability and funding model for the platform. And this could range from support from different institutions to national research councils to potentially support directly from government departments. And I've put a few links on the right-hand side of this presentation, which I'll make available to everyone at the end, so you can, for a bit of further reading on these topics. So I wanted to briefly discuss some examples, some national examples of backbones and biodiversity portals. So I'll start with one close to home, and I have to freely admit that the UK is perhaps not the best example as a reference country for taxonomic backbones. We use a very simple national checklist. We do have the National Biodiversity Network, which includes a comprehensive national atlas. So for us, this is set up as a charity, and much of the national monitoring is done by a volunteer network. And as many of you will know, the UK has got a rich history of volunteer natural historians going back for over 100 years. Our names are fairly well known. We're not a mega diverse country, and it's fairly stable. But it's worth mentioning that our national atlas is based on the Atlas of Living Australia software, and this is free and open source biodiversity enterprise scale platform, which has been made available to other countries in a community program convened by GBIF, which continues to maintain and extend this platform's functionality. It's now used by more than 20 countries around the world. So Australia, as previously mentioned, has a national biodiversity database, the Atlas of Living Australia. It's important to note that they don't use the Atlas as an authority for managing species names directly. They devolve this to 5 national species lists or backbones, and also make extensive use of 2 international lists or name backbones, the Catalogue of Life and the World Register of Marine Species. So moving on to additional kind of mega diverse countries. So Brazil have got a really good national biodiversity database, but I would say that their Reflora project is probably of most interest to Malaysia as a potential model, or at least on a focus group. So they have a government-supported and international effort to create a national flora and a virtual herbarium, including collaborative digitisation, similar to the project with LIM, but on a larger scale with herbaria around the world. And finally, Colombia is probably or possibly the best national model for a mega biodiverse tropical country. They have a highly coordinated national effort across taxa, many participating institutes, new national legislation that really strongly supports this new digital work, and are a very active GBIF node. So lots of good lessons to be learned there. And just as a few quick examples, so this is their GBIF hosted portal where you can see that they're listing biological records, lists of species, photographs of species, publishers, and links to all Colombian diversity data sets on GBIF. And each checklist can be maintained by separate publishers and contributors. So these are listed here. And each of these gets assigned a DOI and is a citable publication in their own right. And in the near future, it will be possible to use GBIF to accommodate national taxonomy, a names backbone, and organize all GBIF records from the country against this single national taxonomy. And that would make it much easier to use and contextualize Malaysian biodiversity records from other collections of projects around the world. And I wanted to end on perhaps a few discussion points that we could touch on today. One of the first is, can you use any existing systems? This could save you time, money, and give quicker results. What are your priority taxa? Are there floristic or faunistic treatments that you can readily digitize to start filling in some of the gaps? And what would you consider quick wins? Would this be data for national reports, feeding into national legislation, checklists for national parks, efforts to support long-term monitoring projects, or perhaps focus conservation efforts? But these are really good things to think about, products or things you would want to get out of a national backbone. And today I'd just like to conclude to say that I'm joined by a couple of colleagues from GBIF, Tim Hirsch and Andrew Rodriguez, who can answer any questions about hosted portals or questions related to GBIF technology. Thank you.
Thank you very much, Mr. Lawrence. I hope you can stay tuned until the end for the panel discussion.
I will, thank you.
All right. Next, we have Mr. Yasser Muhammad Arifin, who will be talking about the updates on MyBees. Mr. Yasser is the Assistant Secretary at the Division of Biodiversity Management, Ministry of Energy and Natural Resources, or KETSA. He's also the system developer for Malaysia Biodiversity Information System, or MyBees. I remember the MyBees team, particularly Mr. Yasser, often faced cold treatment from scientists who are at first very suspicious of your intent or your cry for help in order to develop MyBees during the early stages. So we are going to listen to Yasser on the updates of MyBees. Please, can you share your slide? Okay, you can have the floor now.
Terima kasih Prof. Nayati. Izinkan saya untuk pentangkan dalam bahasa kerana ia lebih memudahkan untuk saya buat penerangan depan ini. Tapi tuan-tuan boleh merujuk kepada slide dalam bahasa Inggeris lah. Okay. So Malaysia Biodiversity Information adalah inisiatif kerajaan dalam mengumpul semua maklumat biodiversity di bawah satu rangkaian lah. Dia berbalik kepada isu apabila kerajaan ini mendapatkan data-data berkaitan biodiversiti, kita mengalami kesulitan untuk mengakses data tersebut lah. Pertama dari segi untuk mengumpul, menganalisis dan juga menilai data tersebut. Jadi kita perlu wujudkan satu data yang centralized. So kat sini adalah punca kuasa yang mengapa kita perlunya ada satu centralized sistem, iaitu bermula daripada CBD sendiri di bawah Convention on Biodiversity, di mana negara yang menyertai CBD perlu mewujudkan satu clearing house mechanism, iaitu CHM, yang digunakan pakai untuk tujuan pelaporan dan juga perkongsian maklumat berkaitan dengan biodiversity. Jadi ini salah satu titik tolak mengapa kita perlunya ada satu sistem yang centralized. Dan sistem ini diletakkan fokus pointnya di bawah Kementerian Tenaga dan Sumber Asli, di bawah pengurusan bahagian biodiversity. Kemudian kita ada punca kuasa kedua, mengapa kita perlunya ada satu bekalan data yang centralized yang disebut oleh Dr. Naim tadi, di mana disebut di dalam National Policy on Biological Diversity, NPBD 2016 dan 2025. Dalam target 16-25 menyebut bahawa perlunya ada satu sistem yang centralized bagi mengumpul maklumat tersebut dan tujuan pelaporan kepada national reporting. Dan yang ketiga, kita perlunya ada satu centralized juga disebut di dalam akta yang baru saja kita riliskan, iaitu access to biological resources and benefit sharing, akta 795 atau ABS, di mana di dalam section 32 menyebut perlunya establish and maintain a clearing house mechanism, terma yang sama digunakan pakai. Jadi menyebut bahawa perlunya ada satu bekalan data yang centralized juga lah, yang akan mengumpul maklumat-maklumat berkaitan biodiversity supaya kita dapat mengawal dari segi akses dan juga kita akan pulang kembali benefit kepada penyumbang sumber biologi tersebut lah. Okay. Di mana dalam akta ini juga menyebut pasal information, kalau kita tengok dalam akta ini menyebut bahawa information itu merujuk kepada semua elemen biological resources, genetic resources, dan juga traditional knowledge juga lah, secara fizikal atau non-fizikal sendiri. Yang ketiga, punca kuasa yang seterusnya adalah di bawah National Action Plan on Invasive Alien Species yang baru saja rilis pada tahun ini 2021 hingga 2025 dalam target goal 1, target 1, activity 1, di mana perlunya ada centralized data berkaitan dengan IAS di dalam National Clearing House Mechanism. Istilah yang sama digunakan pakai ialah di mana pihak Sekretariat IAS Kebangsaan telah bersetuju untuk mengangkat MyBis sebagai bekalan data bagi maklumat-maklumat berkaitan dengan invasive alien species. Sedikit saya nak beri penerangan kepada tuan-puan. Di bawah CBD sebenarnya kita perlu menyediakan 3 clearing house, iaitu National Biodiversity Clearing House yang kita kenali di Malaysia sebagai MyBis. Kita rebrandkan di atas nama MyBis supaya lebih jelas lah istilah tu. Kalau kita guna nama clearing house tu agak keliru. Mungkin public tak berapa faham apa itu clearing house. Mungkin dia ingat bersihkan rumah ke something itu. Jadi kita tukar istilah tu tersebut kepada yang lebih spesifik. Dan National Biodiversity Clearing House merujuk kepada ACI Biodiversity Target. Yang kedua, kita perlunya ada Access and Benefit Sharing Clearing House yang kita wujudkan di bawah sistem yang sama, di bawah architecture yang sama, di bawah MyBis, iaitu kita panggil MyABS yang merujuk kepada Nagoya Protocol. Dan yang ketiga adalah berkaitan dengan Biosafety Clearing House, Biosafety CHM, yang merujuk pada Kategana Protocol. Dan yang ketiga, biosafety ini terletak di bawah bidang kuasa Jabatan Biokeselamatan, iaitu di bawah Kementerian KASA, Kementerian Air dan Alam Sekitar. Dan yang keempat pula lebih kepada penambahan kepada CBD juga, iaitu di mana Malaysia terlibat dalam LMC, Like-Minded Mega-Diverse Country. Jadi kita pun menyediakan sistem pemantauan pelaporan ke LMC di tingkat antarabangsa. Sedikit objektif berkaitan dengan MyBis. Tujuan kita buat pekalan data ni bukan sahaja untuk kita merekodkan data. Kita juga bertujuan untuk memberi akses kepada data tersebut lah. Dan juga kita nak wujudkan public awareness kepada semua masyarakat lah berkaitan dengan kepentingan pemeliharaan dan sumber biologi kita. Dan pada akhirnya data-data yang dikumpul kita boleh sediakan statistik untuk analisis dan kualiti data. Sedikit progres terkini, pada tahun 2016 kita menang Gold Award untuk tandingan clearing house mechanism di tingkat CBD, di mana 190 negara menyertai pertandingan tersebut dan kita diiktiraf pada negara new national yang berjaya menyediakan greenhouse yang terbaik lah. Dan pada tahun 2018 kita telah mendapat Certificate of Achievement di mana kita berjaya mengekalkan kualiti pengalaman data CHM kita betul terus antara top 13 dengan top 14, dan untuk Certificate of Achievement ni hanya 4 negara saja yang menerima pada top 14 tersebut lah. Dan yang terkini pada tahun lepas kita telah mendapat pengiktirafan daripada Malaysia Book of Record bahawa kita merupakan pekanan data yang terbesar sekali lah yang berkaitan dengan biodiversity yang kita berjaya kumpulkan. Dan berikutnya adalah sedikit statistik ada terkini berkaitan dengan pekanan data biodiversity. Yang pertama, kita telah mengumpulkan lebih daripada 47,000 spesies berdaftar di dalam pekanan database kita. Yang kedua, kita telah merekodkan lebih daripada 128,000 specimen dengan kerjasama agensi-agensi yang mempunyai pusat koleksi. Dan saya lihat kebanyakan peserta panelis dan juga yang hadirin sekalian pun pernah terlibat dalam program ini. Dan seterusnya kita telah digitalkan lebih kurang 12,000 spesimen, termasuk juga dengan projek NHM London di bawah Mr. Lim, di mana kita merupakan pekanan data untuk menyimpan data-data digital spesimen tersebut lah. Untuk foto pula kita telah merekodkan lebih daripada 11,000 gambar-gambar spesies. Kalau kita lihat kebanyakannya masyarakat umum ni dia lebih tertarik tentang biodiversity apabila kita menunjukkan gambar-gambar. Kalau tuan-tuan boleh tengok gambar seperti gambar burung yang berwarna hijau tu, jarang kita lihat. Jadi kalau kita letak gambar tersebut mungkin akan menarik minat orang untuk mengenali tentang biodiversity. Kemudian kita telah merekodkan lebih daripada 400 pakar di Malaysia. Kalau untuk pengetahuan tuan-tuan semua, MyBiz juga merupakan directory kepada pakar-pakar biodiversity di Malaysia lah. Mana kita mendaftarkan semua pakar-pakar di dalam satu directory yang tuan-puan boleh cari sendiri pakar-pakar tersebut dan berhubung dengan pakar tersebut jika ada isu-isu berkaitan dengan biodiversity. Dan pakar-pakar tersebut bukan saja dalam bidang biodiversity, kita juga linkkan juga pakar-pakar yang luar daripada bidang biodiversity. Contohnya pakar-pakar dari segi law, undang-undang, pakar-pakar dari segi teknologi, IT, yang kita linkkan ada related dengan biodiversity. Dan kita dah rekodkan lebih daripada 4,000 sumber rujukan yang telah diterbitkan berkaitan dengan biodiversity di Malaysia. Jadi semua bahan-bahan yang didaftarkan di dalam pekanan database kita memerlukan reference. Jadi data-data tersebut bukannya data-data yang muncul secara tiba-tiba. Dia mesti ada rujukan yang diterbitkan sekali. Dan juga kita telah merekodkan lebih daripada 400 kawasan perlindungan di Malaysia. Mungkin saya akan terang lebih lanjut tentang pada slide berikutnya. Untuk yang bawah tu pula adalah negara-negara yang mengunjungi dan melihat kita punya data tersebut lah. Setakat ini kita tidak ada lagi kerjasama untuk dari segi mengambil atau menggunakan data tersebut. Iaitu daripada Indonesia dengan negara jiran kita, Indonesia, Thailand, Singapura, Jepun, Filipina. Mereka sangat berminat dengan sistem yang kita bangunkan kerana mereka pun belum ada lagi sistem established sepenuhnya seperti mana yang kita telah laksanakan. Kemudian negara-negara jauh sikit daripada serantau kita, daripada China, India, Australia, UK dan US sendiri lah yang melihat pada data-data kita. Sedikit tulang belakang daripada pekanan data biodiversity. Species profile merupakan tulang belakang, backbone kepada kita punya pekanan data lah. Bila kita sebut satu pekanan data, kita mesti tahu dia punya struktur asas dia. Jadi bila kita sebut biodiversity, species profile adalah struktur asas kepada pekanan data. Kalau tuan-puan tengok slide pembentangan oleh panel sebelum ini, dia lebih kepada spesifik taxonomy. Tapi kita untuk pekanan data biodiversity, kita spesifik kepada species. Sebab daripada maklumat spesies itulah kita boleh berkembangkan berbagai-bagai lagi data seperti maklumat taxonomy, dia punya description, foto, habitat, even termasuk juga IUS status, location, specimen collection, expert yang tahu tentang spesies tersebut, dan banyak lagi yang kita boleh perkembangkan. Dan seterusnya adalah modul yang penting juga dalam kita punya pengolahan data, iaitu modul specimen yang merupakan pengukuh kepada maklumat spesies profil tersebut. So, macam mana kita nak buktikan bahawa spesies tersebut berada di Malaysia adalah melalui maklumat spesimen ni lah. Jadi kita perlu adakan satu juga rekod untuk menyimpan maklumat spesimen tersebut. Di mana kementerian sendiri di bawah KEXA telah mengajukan satu program yang kita panggil program inventory spesimen kebangsaan. Di mana pada permulaan pada tahun 2016 saya Prof sendiri pun terlibat dalam program tersebut dalam merangka struktur modul untuk pembangunan modul spesimen ni lah. Dan juga
I'm sorry, Yasir, you have to wrap up soon.
Oh, okay, sorry. Okay. And then untuk pengetahuan semua, kita telah merekodkan lebih kurang 128 ribu tadi lah. Daripada program tersebut kita telah melibatkan 14 agensi yang mempunyai pusat koleksi. So ini berkaitan sikit tentang protected area, di mana Malaysia MyBis merupakan pengolahan data untuk merekodkan semua maklumat protected area lah. Mana data-data tersebut kita submit kepada WDPA sebagai data kebangsaan. Dan juga kita telah merekodkan lebih daripada 400 pakar yang saya cakap tadi yang terlibat dalam program ini, dan juga mereka merupakan pakar-pakar yang akan validate data-data yang berada di dalam pengolahan data. Dan juga dari pakar tersebutlah kita boleh buat berbagai program lain selain seperti penerbitan dan juga research lah. Okay, sedikit produk yang hasil bila kita dah kumpulkan kebanyakan data ni, kita dapat wujudkan beberapa produk lah, iaitu yang pertama MyBis API dan MyBis SSN, di mana API ini boleh digunakan pakai oleh sistem-sistem yang ada di peringkat agensi ataupun di peringkat universiti. Data-data MyBis itu boleh di-linkkan melalui API yang kita sediakan melalui SSN number lah, iaitu Unique Identification ID, di mana hanya merekodkan nombor ID kita boleh dapatkan lebih detail tentang spesies tersebut lah. Dan maklumat API dan SSN boleh digunakan dalam penerbitan kewajipan dan juga dalam maklumat spesimen. Sedikit MyBis QR dan MyBis Bot, di mana kita boleh mewujudkan QR code bagi setiap spesies profil dan juga spesimen profil. Bila kita dah satu pengolahan data, dan juga bot untuk kita buat sistem integrated, di mana tuan-puan hanya perlu buat carian melalui spesies ni menggunakan aplikasi yang sedia ada dalam tuan-puan. Seperti contoh yang kita pernah integrated iaitu dalam Telegram, di mana tuan-puan boleh search sendiri maklumat spesies melalui Telegram aplikasi. So untuk MyBis QR, kita dah telah bersama dengan beberapa botanical garden. Contohnya kalau tuan-puan pergi kepada Pusat Botanical Garden, tuan-puan boleh nampak QR code daripada MyBis lah. So ini beberapa hasil daripada produk yang kita kumpulkan. Jadi kita dapat terbitkan beberapa buku yang berkaitan dengan data-data yang kita kumpulkan. Jadi tuan-puan mesti pernah melihat buku ini, dan untuk akses pada data tersebut, tuan-puan boleh sendiri pergi kepada yang public library dan sekolah. So ini collaboration.
Yasir, I think you have to stop.
Okay. Sikit lagi. Yeah.
Okay,
collaboration dengan agensi-agensi yang terlibat, di mana kita telah bersama, dan kami masih lagi senamanya mencari collaboration yang ingin terlibat dalam pengolahan data MyBis ni. So ini program SEPA yang kita telah anjurkan sepanjang tempoh program. Dan sikit future development, di mana kita ke arah untuk membuat satu sistem untuk ABS, Taxonomy Data Information untuk kita sediakan red list punya data, citizen science dan juga specimen digital image. So, saya rasa itu saja saya punya pembentangan untuk hari ini. Terima kasih, Prof.
Okay, thank you, Encik Yasir. Next, we have Dr. Nurzatil Sharliza, who is going to present the topic Fair Data Stewardship for Biodiversity. Dr. Nurzatil is a senior lecturer at Center for Research Biotechnology for Agriculture, or CEBAR, University of Malaya. She's also the principal investigator for Fair Data Stewardship Guidelines for Biodiversity, which is a project funded by the International Science Council, Regional Office for Asia and the Pacific, and the Academy of Sciences Malaysia. You can carry on with sharing your slide, and you can have the floor. Thank you.
Sure. Okay, let me just start sharing my screen. Okay. However, I can see now my slides. Thank you very much, Prof. Hayati, the moderator, and also Dr. Maslan for inviting me to be speaking about a project that the UM team, University Malaya, my colleagues and myself, we just started actually working on this project we call as the Fair Data Stewardship for Biodiversity Research. So I'm taking this opportunity to speak about the project that we are working on and looking forward to hear feedback from all of the participants and panelists. So just before we start, maybe it's good just, let me just introduce you what FAIR means and why FAIR data is important. So FAIR actually is the abbreviation for findable, accessible, interoperable, and reusable. So this is a common principle being used in concept of open science. So if some of you might have heard about open science, so it's all about sharing research data, but of course it's under certain conditions and terms. So when we call data is fair, this means that data should be as open as possible and as close as necessary. In fact, if you listen to the keynote speech delivered by Dr. Naim today, FAIR principle actually is adopted by the National Initiative, Malaysia Open Science Platform, which is spearheaded by the Ministry of Science, Technology and Innovation, MOSTI. So why we believe that FAIR data is important and why we believe that this project is relevant, because we see that there's an urgent need to collect, preserve and share FAIR data on biodiversity. So that goes to different examples of data, in fact, from ecological, species, and also genetic data. This actually to allow for different types of analysis of biodiversity data by different stakeholders, and that includes researchers and also policymakers. So just an overview of the project. Okay, the project that we're working on is a year project. We just actually recently started this month, and we hope this project will end by end of April next year. And we are so fortunate, very thankful to the funder, IC Europe, and also the Academy of Sciences Malaysia, who provided us a seed fund actually to allow for the project to kickstart. And just a bit of background to the project. So when we crafted the idea of this project, actually we see there are issues with biodiversity research, and I think a lot of these issues actually have been highlighted and mentioned by the previous panelists. So really one of the key issues with biodiversity research is lacking in terms of reproducibility, because data poorly managed, archived, curated, integrated, shared, and preserved. So the idea, so one of the strategy that we propose is actually we want to address the curation issue. So we propose to help in terms of strengthening the data stewardship support for the biodiversity data journey throughout the data life cycle. So starting from the data acquisition, processing, analysis, curation, sharing, and also to reuse. So the essence is really what we want to target is we want to make sure that once we are gearing towards the digitization of data and research data sets, we want to make sure that all these data are actually fit, high quality. So when you put a data on a repositories, on a system or repository, the data must be findable, accessible, and interoperable, and also reusable. So that's why this project is proposed. So there are actually three objectives of the project. The first one, actually, we want to develop the hierarchies and rules. We want to understand and streamline the process of the flow of data journey, as well as mapping out the roles of data stewardship in a data life cycle. Then, of course, we want to develop a landscape assessment that will map out the data requirements that meet the researchers' needs, and also to identify the data that they are producing in respective research disciplines, as well as to create awareness and evaluate data stewardship readiness. And finally, and I believe is the most important objective of the project is to actually establish sets of biodiversity data stewardship guidelines for researchers, data stewards, and IT technologists. So to achieve the objectives, really, we actually are backed by the strong team members. We actually have also Mr. Lim together with us in the team. We also have different researchers from multiple backgrounds. We have the taxonomists, we have geneticists, we also have biodiversity, informatics, and also IT tech person, stewardship piece person, just to brainstorm together to make sure that this project will deliver what is promised. So when we actually started, this month, May, okay, and we actually will start with doing some groundwork, a lot of benchmarking, that study, and also workshops, just to make sure that we collect to scope to do the scoping of our project of the guidelines, and also do some surveys just to identify what are the issues that researchers are facing in respect to the data life cycle, in respect of the data sharing, and then of course after a few months later we'll follow by the establishment of the working groups. So we're going to get some experts, in particularly the data holders, the data generators, to help us steer the direction of the guidelines, and also provide us inputs in terms of the guidelines. And finally, for the next 6 months, we're going to do a lot more engagements with stakeholders to craft the guidelines for the fair biodata stewardships for researchers, data stewards, and IT technologists. So we expect that the whole process will complete in a year. So by the end of the project, which will be April 2022, we hope that we will come up with sets of biodiversity data stewardship guidelines. So some of the aspects that we see that will be covered, will be discussed during the workshops. We include something like the metadata, all the standards, the identifiers, even data management plan. So those are the things that we will discuss throughout the engagements and the workshops. And again, this set of these guidelines actually will set out the best practices that will cover all data stewardship aspects. So we're looking into policies, procedures, technical know-how. So all this will be relevant and applicable to researchers. So they could be the ecologists, taxonomists, the curators, even the geneticists, and also the data stewards. So they could be the librarians who are trained to be the data stewards, or even the researchers themselves. And also to the IT technologists, who really they are the data custodian of the research data sets. So so as a way forward for this project, so again, we are really in the beginning of the beginning of the project, but we see that this project is high value. We hope that we will provide and lay the foundation towards the open science journey in Malaysia in particular. Okay, so this project will set the foundation towards the digitization of the record of specimens and even the research data sets. And towards that, of course, these guidelines will help in terms of the giving ideas, what sort of training is needed for the domain specialists, biodiversity data stewards, and also contribute towards the efforts of digitizing the biodiversity specimens. And of course, towards the end is we want to connect and share among the communities, especially the researchers, And we see that there's a huge potential gearing up towards the Malaysia taxonomy backbone. So for any other questions, or if you're interested to be part of the workshop participants that we're going to conduct in the next few months, please feel free to contact me directly, and also our project analyst, Ms. Suhaila. And we'll get back in touch with you soon. Okay, I think that's all from me, Prof. Thank you very much.
Thank you, Dr. Nozatil. Okay, we have now come to an end on the presentation by the panelists, and the most important time is now the panel discussion. I'm sure there are many questions already posted by the attendees. We are going, there are 45 minutes for this session. And I think many people posted about the same question to at least one of the panelists. So I'm going to start off this session with the most burning question to Dr. Khairul Naim. If you are still around to answer the question, Dr. Khairul Naim. Everybody, I'm sure many of us wants to know what happened to the idea of having our very own national natural history museum. Can we have Dr. Naim? Otherwise, I think.
Sorry, Yati. I think maybe Yasir will be able to answer.
Yes, since Yasir is also in the same group.
Yes.
Same team with
Right,
Dr. Naim.
Yes, I think so.
Mungkin saya boleh bercerita sedikit lah mengenai hal tuju pembangunan NHM di Malaysia. Sebab tu yang menjawabnya adalah Dr. Naim lah yang lebih tepat. Cuma apa yang saya maklum bahawa kita sekarang dalam perancangan untuk mewujudkan NHM di Malaysia. Dengan setelah penubuhan NBC, ya, dan di mana pengurusan NHM tersebut akan berada di bawah NBC. Dan perancangan kita untuk lokasi NHM tersebut adalah di Putrajaya. So perancangan masih lagi di peringkat kementerian untuk finaliskan.
Many people confused between NBC and the proposed Natural History Museum. So these two are separate, two different things. And while we are still waiting for the for most of the wishes of all taxonomists in Malaysia to have our very own natural history museum, well, at least we can fall back to the NBC on matters pertaining to the database of biodiversity of Malaysia. Maybe I would like to throw a question to Dr. Liu on taxonomy, because you know, when we talk about taxonomists, people who the actual people who work on the ground, maybe a round figure that the world has is probably more than 10,000 taxonomists worldwide, which may seem like a big number, but when there are about 15,000 new species of all types being logged each year, suddenly we are looking at a global crisis, or rather a taxonomist crisis. So in order to train one would probably take 5 to 10 years. And being a taxonomist yourself, what do you think the situation in Malaysia is like?
So thank you very much for the questions. I don't see myself as a taxonomist, by profession. Just by chance that when I study the biodiversity, there are some species that without name, so happen to name a few species. But to go back to the question, actually I did some calculation on this many years ago, 10 years ago. So so let's say given the current pace, how we describe the species, it will take about 400 years to describe everything on Earth. 400 years. So it's about 10 generations of taxonomists. So we assume that all the taxonomists live long enough to complete their career. So it will take about 10 generations. So I think the point here is just to make sure all the information is accessible, and then let the public to access the information. I don't think the current.
Can you hear me?
Yes.
Yes. Okay. So I don't think the current way to train the taxonomy is still sustainable, because there only a limited research institution available in Malaysia, and they cannot give the job for all the people who are interested in taxonomy. So I think we need to
Testing. Prof, can you hear me?
Yes. Yeah. Currently, we are listening to Dr. Leo, and we'll get back to you for your question later after this. Dr. Naim. Okay. Sorry, Dr. Leo, can you carry on?
Okay. So I think the best way actually is to make the information available, so that any layman, any kids can immediately start to work as a taxonomist, to access all the information, to reduce all the barrier, to help them to sustain their curiosity, to be interested in the biodiversity. I think this is a way to move forward. I don't think we still can rely on the trend taxonomy to be sit in the institution and solve the taxonomy problem. As we can see in overseas, there are a lot of the taxonomists, amateur taxonomists who are quite competent. They have the daily job as a postman, as an engineer, but they can do a very good taxonomy work, right? So I think this is a way forward. We just need to make sure all our knowledge is shared. If for those result is conclusive, we can publish. Even during the process, like what I shared just now, even all our note or everything, we can just share it through the facility that are available. Yeah.
Okay, but don't you think that Malaysia falls back a little on having, you know, the naturalist among the public, but do you think that choosing this field of taxonomy as your career is because it's not very lucrative? Simply put, there's no money in it. You can't get rich being a taxonomist. For example, if you were trained as a mycologist, you wouldn't choose to work in a research institute or university, but rather maybe work for Carlsberg, for example, or Kraft or any other big industry. So how can we encourage young scientists to become taxonomists in research institutions or universities?
Yeah. So I think, for example, the apps that I show you, the iNaturalist, I think this is a very good app to get the kids to be involved. I think every kid at any moment of their life, when they encounter insect or animal, they will have the curiosity. But the problem is there's no facility that can sustain and then let their curiosity to be expanded. When they see some very interesting beetles, for example, but they cannot know the name, then the curiosity just stops there. So I think this is also related to our education in science, actually. So I think we need to, back to the point, I think still we need to share the knowledge. Just share everything that we have. Just share it, as simple as that. Yeah. Let the kids to discover what is available. Now they are very competent with the tools on their hand. Just a few clicks, they can get the information that they have. And make sure we use the platform that are very easily accessible. Don't require them to go through, if possible, just one click to get everything that they need. Yeah, I think. Yeah.
May I ask, where do you keep your specimens? Because I know that Borneansis collection is among the best depository center for in Malaysia. Do you have any problem or challenges in specimen upkeep?
Okay, so
you know, being the best center in Malaysia, do you still have problems?
Of course, we have a lot of problems. The main problem is a lot of people have a mindset that all the specimens must be identified before we catalog them. So, and then at the same time, there's not enough taxonomists in our country, which a lot of the specimens left unidentified. When they are identified, they didn't catalog them. That's a problem. As time go by, this uncatalogued, identified specimens have been accumulated, and then this takes a lot of the effort to digitize them, then key in the data. So actually, for important seeds, at least for the snail mollusca collection, we adopt an approach in which we make use of the database. We have the in-house developed database in our institution, in our institute. We use the database as our daily workflow. We never key in data, we never upload image, but we use it as a workflow. Whenever we need to enter information, we will enter in the database. We will not use other platform. So we try to integrate the database in our workflow so that whenever we finish the work, all the data is available. We don't need to think about whether we need to digitize the data or we want to key in the data. So I think this is what is lacking in most of the institutions in Malaysia, which they do not integrate the database in their workflow, but they see a database for them to store data, to key in the information, to upload the information. Yeah. So it seems like a redundant work for many researchers, including taxonomists, to why I need to key in the data. I keep the data in my Microsoft Word and Excel. I'll be more than happy enough. Because they use the Microsoft Word and Excel as their part of their workflow. They never use the database. So I think what is needed actually is to need to have a database, not top down, but bottom up. We need a database that allows the researcher to be used when they do their research. Not after they finish their research, then we give them database, you have to key in data. This is too late, I think. Yeah, thank you.
Okay, thank you for your response, Dr. Liu. Can we have Dr. Naim?
Yes, Prof. Sorry for
Naim.
some technical glitches. I have to use my handphone to answer the burning questions that I received from a number of our participants, Prof. Tan Sri Saleh, Prof. Ahmad Ibrahim, Dr. Nur Salwa on the Malaysian Biodiversity and National Natural History Museum. I fully agree with Tan Sri and Prof that establishing NHM is a bold step and a game changer, Prof, in improving our Malaysia's biodiversity management. And as all of us aware, Malaysia, although one of the richest countries in biodiversity, is only the few countries without NHM. If we look back at the decision of establishing MBC, it was made actually as early as in 2006 at the meeting of National Biodiversity and Biotechnology Council, where it was decided that there is a need for Malaysia to establish NHM. So many initiatives have been taken to realize the establishment. But as of now, its establishment has yet to be realized. For your information also, KEXA has established core team of Malaysian Biodiversity Center with 5 personnel to spearhead the establishment of NHM. MBC will play a greater role, and it will be the umbrella for NHM and other key functions in providing advisory for technical policy and science interface, managing database and repository, also managing funding for various conservation activities, playing a key role in implementing national competent authority for access for benefit sharing, etc. Fortunately, we have identified the locations, but I couldn't reveal at this moment. We have also finalized the proposal and we have submitted the proposal to respective agencies for further consideration. So we really hope that we can realize MBC very soon, Prof.
Okay. Thank you, Dr. Naim, for your response. I hope everybody, I'm sure there are other questions pertaining to our Natural History Museum. And previously, the problem lies in the name, you know, because having the name museum there would conflict with the museum, Jabatan Museum Negara. But you know, I think we can sacrifice the name museum and have it in a different form, or having a different name, just to have the Natural History Center, or whatever, you know, come into fruition. Another question, I think this would be interesting, that maybe Mr. Lim can answer. You know, how about the wet specimens? I mean, so far, you've handled only the dried specimens. What about the wet specimens or big specimens? Can you easily digitize those specimens?
I think Lawrence is best to answer this, because I asked him this question when I was there. I said, all these jars, how do you digitize them, you know? So I was at the marine collection, the amphibians, you know. So the marine invertebrate is a different ballgame altogether. So I would like Lawrence to maybe handle this question. He knows best, okay?
So it is possible, of course, to digitize some of these liquid preserved specimens. In the previous collaboration with Lim and colleagues, we focused on pinned insects and herbarium sheets because they're quicker and cheaper to digitize. So in terms of species coverage, that was an easier focus. But of course, it is possible. It just takes a bit longer, and you have additional concerns if you're handling specimens preserved in methanol or formaldehyde. But we have set up to digitize those, and if you look on our data portal, and indeed on some of the specimens that we've shared with Lim, that includes some liquid preserved specimens. And in one of the questions in chat, I've also shared a paper that some of my colleagues on a previous project prepared that talked about the state-of-the-art practices for digitizing liquid preserved specimens. And we're also working on a project to digitize a couple of families of horseshoe bats and relatives, which is a kind of COVID-related research project that we're doing, which will be made openly available later in the year. And that includes some imaging of the jars. So it depends what you're doing the project, whether you're looking for the label data or you're looking to for the specimens, because you know it varies depending on what you want out of the research, but it's certainly possible. It's just a bit slower than herbarium sheets.
Okay, thank you, Lawrence, for your response. And here I have a question by Ivy Wong, but I think Dr. Naim has already resolved or answered this question. Have the ministries resolved the ownership by MOTAC to the word museum? That was the stumbling block previously in setting up the Malaysian Natural History Museum. I don't think there is a big issue right now, because anything regarding the proposed Natural History Museum is now going to be handled by the NBC. And we look forward to the progress or more news, especially on the location of the physical Natural History Museum in Putrajaya, where the location is.
Prof, if I may, Prof.
Yes.
Just a short response to Miss Ivy. Actually, it is just some jurisdictions issues, but I think it can be resolved by put national first. Yeah, Prof. That's all.
Okay. Here's a question, an interesting question. Perhaps Mr. Lim or Lawrence can answer also. What is the next level of digitization of specimens? Have we gone into 3D yet?
You're
Okay,
muted,
sorry.
Lim.
Can we actually produce 3D version, digital of specimens?
Yes, we can. So in NHM London, we've got some really good micro CT, scanning electron microscopes, which can produce 3D images of specimens. And I was replying to one of the questions in chat about digital repatriation. We're hoping to extend more digital services, so beyond just two-dimensional photography of our specimens, we're hoping to also include molecular requests as well for some of our tissue samples. So in the future, we want to be able to make 3D imaging either through CT scanning or electron microscopy available to external researchers. And in fact, over COVID, it's been really interesting because a lot of our light microscopes and electron microscopes we've set them up so that they can be remotely controlled by researchers. And so we see that as potentially a really powerful thing because, of course, for researchers in Malaysia, you could potentially use these machines when we're all asleep, which at the moment they're going unused. So we're thinking, how can we make these analytical facilities more available to external researchers? But yes, 3D imaging can be done. It does pose some more technical challenges if you're trying to host the data, but a number of institutes are doing it, and I'll put some links to CT scans and 3D scans in chat in a bit.
If I may, Prof. Nohayati.
Yes.
You know, I mean, after coming back from London, right? So I actually tested, you know, their method of, imaging and how little resources are required to do a nice collection. So what I did was that I worked with Dr. Henry Barlow, who has a very big, huge moth collection. Okay, his Genting Tea Estate, I think, contain one of the most complete moth collection in the Klang Valley, I would say. Okay, he gave me a project. He says, he has only one assistant, a master student, and himself. Okay, and with technology, can we scan 18,000 specimens within a year or 2 years? And actually, we finished the project within that 2 years. So I've given the links here on the web, on the chat here. Okay, the Moths of Borneo, webmothsofborneo.com, and a separate project. We also work together with one of his other project based in Old Sabah, the Pyralis of Borneo. That one is a more specific publication, but it's no less less challenging. It's about 1,000 specimens of the thyroididoid. Okay, that one, it requires us to generate the QR code to each of the specimen pages. Now, what I did was nothing great. What we did was we just took the basic process from London, replicate locally, and we see how many people is required to digitize this body of information. You'll be surprised, very few people. But the process has to be clear, has to be precise, okay, and it has to be done again and again and again. And once the benchmark is set, the quality is very good. So I believe what we need at this stage, even though we don't have our own NHM, we need to really streamline our processes. Okay, get together not only just taxonomists, also get together with the computer guys, the IT fellas, the database fellas. Put two minds together. Collaborate to come up with a very, very efficient system. We don't have many people, like Dr. Liu said. We don't have many people. He does not need to do all the database work. Why he has to do that? Let the computer guy do. Okay. But if we can collaborate like this, a lot of things can be done. A lot of things can be done. Okay.
Yes, because classical taxonomists usually work in silos or work alone.
Yes,
but then nowadays it's a trend towards team thinking or teamwork.
Yes.
So I think having a teamwork can actually encourage more innovation or can produce more ideas for innovations.
Right.
Okay, I think we have some more time for some question. Maybe for Dr. Nurzatil, I have a question for you.
Yes.
There's in Europe, there's a new project called a Red List of Insect Experts in Europe. And this question was posed by Ms. Wan Farida Wanjuso. Okay. Have we started on an inventory of Malaysian experts? And or what's the current trend of taxonomic experts in Malaysia?
Taxonomy
Or
expert.
if
Not sure if I'm the best person to answer this question, to be honest.
Or maybe Yase can help.
Yeah.
Do you think we need a red list of taxonomists in Malaysia?
But to me, in my personal opinion, I think it's a useful list. I mean, like the work that we are doing at UM at the moment, we are actually gathering all these experts, and that also includes taxonomists. So once we have these people, once we bring these people together, so we actually have kind of like started the list of experts in taxonomies. And of course, when we have the senior taxonomists coming in, we are also going to invite the junior taxonomists joining. So we can have the kind of like, whether we want to call it as a training or mentoring or knowledge exchange. I think this is one way forward to ensure that taxonomy field, biodiversity taxonomy field will, have dia punya apa kata kelangsungan kan, the continuity of it. Yeah.
Okay. And do I have time for another question? Maybe another question about the database or the software, because many institutions in Malaysia use a particular database software called BRAMS. And for example, UCAM Herbarium used to use this software because it is connected to Kew Garden, and therefore is to make sharing information more easier. But then since the staff who was trained to handle this software unfortunately died, and we wasn't able to get a replacement, the herbarium is back to using Excel. And the system used to be freely available, but now you have to subscribe, and it's quite expensive. I think around 20,000 ringgit. And you have to retrain, you have to train somebody. And I think FRIM provides the training. Are there what are the alternatives? Do you see the future for a web-based database, a single consolidated, which is what we are trying to achieve now? Can MyBiz take upon this responsibility? Maybe Yase or Dr. Naim boleh, can you respond to this question?
Mungkin saya boleh respon sikit berkaitan dengan sistem yang ada di Malaysia ni. Sebenarnya kita tiada halangan ya untuk agensi atau universiti menggunakan mana-mana sistem dalam pengurusan, tetapi kita perlu ada satu yang standardized, yang seragam untuk kita pastikan maklumat tu boleh dikongsi dan juga integrated online. Yang pentingnya bila kita menggunakan satu architecture teknologi pengkalan data ni, kita kena pastikan kita memenuhi standard tu lah. So, seperti mana yang kita tahu bahawa data-data berkaitan biodiversity ni kita menggunakan Darwin Core sebagai standard dalam penyeragaman data tersebut, metadata tersebut lah. Jadi bagi saya yang kedua yang penting kita lihat adalah dari segi sustainable pada sistem tersebut ya. Macam mana kita tahu kita mengikutkan dengan keuangan ya, untuk pastikan sistem terus dipakai, juga melibatkan kos dari segi maintenance hardware ya, dari segi tenaga kerja untuk menguruskan. Jadi kita perlukan satu sistem yang fleksibel dengan keadaan semasa, dan kita tak tahu pada akan datang mungkin teknologi telah berubah dan benda ini akan berubah ke arah perkara yang baru yang kita alami sekarang. Jadi bagi Kementerian sebenarnya kami tak ada halangan dalam pengurusan, tapi kita boleh menyediakan ya. Kebanyakannya yang kami buat program inventory ni adalah untuk membantu pusat koleksi yang belum ada sistem. Itu yang kita cuba bantu. Sebab Kementerian lihat masalah ini hanya boleh diatasi dengan kerjasama tersebut lah. Mana kita menjadikan platform dan juga peruntukan keuangan bagi memastikan pengurusan data kita teratur dan kemas. Itu dari saya. Terima kasih.
Okay. Maybe Yasir, can you continue? Remember initially in the beginning, you found that it's very difficult to get the scientists to collaborate or to help MyBees. Do you still face the same problem?
Okay. Setakat ini kita belum pada permulaannya kita menghadapi kenkangan tersebut. Isu yang utama adalah bila berlakunya perubahan dari segi pengurusan data itulah, daripada kaedah manual ke kaedah digital kita mengalami banyak kenkangan terutama dari segi mengubah mindset minda penyelidik kita supaya menggunakan sistem dalam pengurusan. Kalau kita lihat pengurusan sebelum ni kebanyakannya mereka menggunakan kaedah manual secara rekodnya. Jadi bila kita membawa kaedah baru ni, kita memerlukan masa kepada mereka untuk learn, belajar semualah. Pada permulaan memang ada halangan, tapi sejak kebelakangan kita menerima feedback yang baik dan juga maklum balas yang baik daripada semua. Terima kasih.
Okay, Mr. Lim, I believe you still maintain Alam, which is an atlas of Malaysian diversity, but you also collaborate with MyBees, especially with the Raffles specimens that you've digitized. So both systems are available online.
So what happened is that Alam is modeled after the Atlas of Living Australia. Okay. So upon the advice of Lawrence, so we just actually basically modeled that for our use. But the thing is that I think we are not there yet in terms of taxonomic backbone is not there, things like that. Many areas is not complete, I would say. We are far behind the Australians in that sense, in the Australian model. Okay. But nevertheless, when I brought back the content, we had to store it somewhere. So we together with Yasin Hassanau funded the project. So we created a cloud service. Now, what Encik Yasir said just now is very important. All databases should have a standard. So when we brought back the standard from NHM, it was basically Darwin Core. Okay. And when I had a meeting with Encik Yasir and his team at the GETSA, all I did was to download everything into Excel file together with the images and pass him the external disk drive. And the next day he uploaded everything into MyBees already. No problem. Zero integration problem. Okay. So I guess this is the critical message that I think Encik Yasir is trying to point just now. Try to make sure any database, whether you're created in a DBase tree or Fox Pro or Excel, whatever you call it, try to make it into Darwin Core. Because Darwin Core has a lot of columns, that you can actually fill in content. So if we do not follow a certain standard, we don't know what content is missing, you see? So once you have the right template, then we know, okay, many information is still not there yet. And when we did the project with the Moth for Borneo, with Dr. Henry Barlow's collection, we insisted, we say it's okay, we use the Darwin Core template. And then we realized, many contents still, many empty spaces, many blanks. So I guess once we have the template correct, the standard accepted, then I think everybody can follow. And when we can interchange, interchange databases, you can store here, store there, and everything fits into place. You don't have to worry about what is the integration issues. Okay, so that was a very important lesson, and I'm very grateful to Encik Yasir. He's emphasizing a lot on Darwin Core, and Lawrence also saying that when you go back, please apply going for in your approach. It can be for any of the 5 areas, zoology, mycology, everybody. So I guess once we have that, then we are set in that sense. Okay.
Okay, thank you. And maybe we have time for the last question, which is, now here anybody can answer. How can the public benefit from the database?
So if I may, so I would say, so iNaturalist has been incredibly popular all around the world, and it's a really nice way. So just last month there was a kind of competition for cities to compete with biodiversity. So the citizens could go out and take photos of nature around them in their garden, their parks, and upload them. And that's been an amazing way for people to engage with biodiversity on their doorstep or in their local or national parks. And I would say feeding in, so if you have a backbone, you can collaborate with some of these big initiatives like iNaturalist. And you don't even have to do anything. They already have this platform there. They already have some really nice mobile apps. All you need to think about is localizing it a bit. So having your national backbone and probably doing translations of the interface. And many of these allow community or kind of group translation, so you can get it into the appropriate languages. So that, I would say, is one key way, a very easy way of engaging the public with biodiversity and supporting some of your aims today.
Okay, thank you, Lawrence. I've seen some local initiatives on developing apps to monitor, for example, roadkills. And I believe we also need to strengthen monitoring among the citizen scientists on the issue of exotic species in Malaysia, because I think the problem has become worse. For example, there are events where somebody, we already have the crocodile snapping turtle in our lake, in our native lake, especially in Putrajaya. So that is worrisome. Of course, these issues, biodiversity, habitat loss, roadkills, exotic species, perhaps another different webinar that we should also organize. Anyway, I think it's time to end the panel discussion. I would like to thank everyone, and I will let Dato Maslan to have the last say in congratulating everybody. But from me, thank you for your cooperation, being wonderful panelists, and also all the attendees who submitted their questions. Thank you very much. I hand over to you, Dato Maslan. Thank you.
Yes. Thank you very much, Yati. So this, I noticed, has been one of the most active interaction between the panelists. They've all replied to the questions by the attendees. And I think there were registered about 250 registered, and I think in the end there were about a little over 200 participants. Okay, I asked you the question, and I asked you, what is your biggest problem in terms of the biodiversity data you have collected? Now, 48% of you said identification was your problem. Now, this is very interesting because I know from talking to Lim, who was involved in naming nature 1, the first one, this was exactly the problem, and that we don't have enough taxonomists. Yati asked everyone, can we make taxonomy more sexy, more interesting to young people? But how do we attract them to this career? It all boils down to, are there opportunities in this career? Will they be able to make a living in this career? Those are the things that we need to address. Similarly, somebody asked about, can we have courses on training for this taxonomy? Then 23% of you said your problem was with digitization. That we had also expected, I think. Now, one of my biggest surprises and sort of made me really panic was the information I got that there are 3 million samples in our data centers that we don't have much information on. And if you really have those 3 million, where are they? What condition are they? And to make matters worse, I got so depressed when I heard from Yati a few days ago that the specimens at UKM, for instance, in the building, the biology building, is in jeopardy because they've had to shut down the building because of issues, mold or fungus, I don't know. So you can imagine what's happening to the biggest insect collection in this region. So I think I got into a bigger panic, but I don't know how to resolve this. But I'm sure it's not happening just at UKM, but others as well. So to me, deposition is also a big question. How do we deposit and all that? And 17% of you had that as a problem. So as Yati says, there are the ways in which we have to go on further by having a few more webinars. And of course, in terms of collection, 11% of you said there's a problem with the collection. Now, this is a good sign because all of you are still going out of the fields collecting those specimens. That is the most exciting part for me. But that's the biggest problem, identification. So where are all the taxonomies? 2, digitization. 3, deposition, where to deposit, and of course with the collection itself. And here we've got to what extent, okay, there's a quick feedback for you. And the last thing that we covered was Yati asked that very, very critical question. What is the benefit to the public with all this bio-D? And Lawrence answered part of it. I wouldn't even go down the path of economy, national heritage, you know. I think that's a done deal. We are all converted here. But I think what we in this community need to be thinking about is citizen science. You know, and he mentioned that project where we got people coming in to deposit pictures and all that. Because if you involve the public in this bio-D, in the realm of a bio-D, that's where we're going to win support from the public. Even they will be clamoring for the National History Museum. So I think citizen science is a big way forward, and maybe we can even have another webinar just to talk about citizen participation in bio-D. But as I said, the success of this meeting, of this webinar is in that we have identified a few ways, a few problems that we need to resolve, and a few ways we need to move forward with. So I'd like to thank again Professor Nohayati for doing this excellent job of moderating this webinar. And thank you for all of you, to all of you panelists, for giving us your passion. I know each of you are driven by different passions, and that's why we chose each of you to give us your insights. But going forward, it is the participants here that will make sure we all move forward in the right way. And I love the chats we've been having. There were questions to the panelists. Some of them have been answered. I would like to invite all panelists to answer those questions offline. So once again, thank you so much for taking part in this. I would like to say here and now that we're going to have another discussion on this. I don't see a lot of the problems we have resolved yet. I'm planning to make some stand on this at the international conference on the tropical science, particularly on bio-D. I don't have all the answers yet, so until I have all those answers, we're going to keep having these webinars. I don't know if there's good news or bad news, but I think it's good news. We will continue to engage you. So ladies and gentlemen, all over the world, thank you so much for your participation today. Please keep in touch. I informally also got in touch with some participants who have made some comments to contact me, and the others panelists have also done the same. Once again, thank you so much for being with us today, and see you next time.
Thank you, everyone.
Okay, thank you, Dato. Thank you, everyone. Take care. Stay safe.
Thank you.
Bye. Bye.
Goodbye.
Bye.
Thanks all. Bye.